things. And where I want to start here today is I want to start talking about the so-called bulk phase digestion. Now, you'll remember that when we drew the intestine, the small intestine, we said that the mucosa was thrown into folds. But even after throwing the mucosa into folds, there's a sizable volume in the center of the intestine. This is going to be the site for bulk phase. Digestion. So when we think about this, we imagine this fluid in here is being churned back and forth and back and forth, because remember, the outer limits of this small intestine is the serosal surface, but right immediately beneath that is smooth muscle. And the smooth muscle can squish the contents of the fluid back and forth and back and forth. So we're going to be mixing this region in here. So the middle of the intestine has the fluid being stirred back and forth, mixed by the smooth muscle. Now, the reason I bring that up is that I want to bring up this image here and spend just a moment on it, reminding you where we have been. So this initial image is the image I just drew on the board that describes the small intestine. So we have this big tube, we have this big open tube, and in the middle of this open tube, the fluid is being squished back and forth and back and forth. And you remember that those folds are composed of these villi, and the villi are multicellular structures. These are made up of enterocytes, thousands and thousands of enterocytes. And what we're going to do today is we're going to divide the region of the small intestine into that bulk phase where things are being squished back and forth, and a very thin layer over the top of these enterocytes known as the unstirred layer. So we're going to divide digestion into these two big areas, into the stirred layer and the unstirred layer. When we do that, what I want to remind you of is this figure. Now, what this figure is trying to show is the relationship of the pancreas to the small intestine. And it shows a couple of things we're going to talk about today. Some of this we've discussed in the past. There are these tubes, basically, which we call ducts that were part of the ducts that were part of the exocrine pancreas. These were a continuous layer of epithelial cells. And some of those epithelial cells were dedicated to binding secretin and releasing bicarbonate. But others were dedicated to binding cholecystokinin. The so-called acinar cells bound cholecystokinin released by eye cells. And that signaled the release of digestive enzymes. And we'll write these out on the board in a minute. But those digestive enzymes were pancreatic amylase, lipase, and the proteases trypsinogen and chymotrypsinogen. They then followed this tube down to a common bile duct and into the small intestine. Now, one of the structures we're going to talk about here later today is this sort of green bag up here. This is the gallbladder. It's basically just a holding site for bile that's been generated by the liver. And then as the cholecystokinin causes the release of digestive enzymes from the acinar cells, the cholecystokinin also binds to smooth muscle on the gallbladder and squirts a little bit of bile into the small intestine. That bile, which has been generated by hepatocytes, is going to be used as a kind of detergent to break up these big globs of fat into much smaller BB-like structures. Yes?
So you said that hepatocytes
Can we hold off on the hepatocytes? We'll come back to the hepatocytes. The hepatocytes we're going to talk about as being the sites that are going to take up glucose and amino acids from the hepatic port of blood. But let's hold off on that because they have two functions. They have the uptake function, but they also have the function to make bile. And we'll try to, at that point, bring together the idea of digestion of the water-soluble molecules of carbohydrate and protein along with the insoluble molecules of fat. And we'll try to bring that all together toward the end. So that's the structure I want you to hold on to. But remember that the acinar cells, the pancreatic acinar cells, these pancreatic acinar cells respond to CCK to release three major kinds of digestive enzymes. The first one is pancreatic amylase. The second one is pancreatic lipase. And the third are the inactive proteases trypsinogen and chymotrypsinogen. So the acinar cells now will release pancreatic amylase. This will now go into the lumen of the small intestine. Its function is to convert starch to maltose. Remember, maltose is a disaccharide made of glucose plus glucose. The function of pancreatic lipase is to convert triglyceride into fatty acids. I should say 2 fatty acids plus 1 monoglyceride. Now, I'll emphasize it here. We'll come back to it toward the end. Pancreatic lipase is an aqueous enzyme. It works only in the aqueous environment, but yet it has to act on this molecule that's largely water insoluble. And so we're going to have a puzzle here of how we're going to have to figure this out. And basically what we're going to see is that this is an enzyme that breaks down triglycerides into fatty acids and monoglyceride, but it really needs some help. And the help is from enzymes called bile. The help is from molecules called bile, which are going to be detergents generated by the hepatocytes. But we'll come back to that at the end. The third character are trypsinogen and chymotrypsinogen. These, as the name implies, the OGEN suffix. These are inactive. Trypsin and chymotrypsinogen, trypsinogen and chymotrypsinogen are activated, I should say converted, to trypsin and chymotrypsinogen. In the small intestine. So again, what these are going to do is in the bulk phase is their function is to convert proteins to peptides. So here in this so-called bulk phase, we find that amylase converts starch to maltose. Here in the bulk phase, chymotrypsin and trypsin convert proteins to peptides. Now, this distinction between a protein and peptide is pretty much definitional. For the most part, we're going to see that the trypsin and chymotrypsin are going to convert big proteins into amino acid sequences that are, 30, 20, 30, 40 amino acids long. So we take these great big long chains and bust them up into smaller chains. That's what we're doing. And then finally, here in the bulk phase, lipase converts triglyceride into 2 molecules of fatty acid and 1 molecule of monoglyceride. So that's going on inside this squished about fluid. It's a storm in here. It's like looking at the water in your washing machine. It's being sloshed back and forth and back and forth. There's a lot of action in here. But there's also this idea of the unstirred. The unstirred layer, which is, 30 to 40 at most micrometers thick, overlying the enterocytes. So again, the villus, let's go back up here and describe a villus. Where the villus is this finger-like projection formed from thousands upon thousands of individual enterocytes. So these are enterocytes of the villus. Then down here, beneath that, let me hash in all of these, will be 2 major structures. And again, to orient you, this is basilar. This is the basal lamina. And then there will be an extension of a capillary that we talked about last time, and I'll show you this in red. And this is meant to be a capillary of the hepatic portal vein. And then immediately beside that, beside the capillary of the hepatic portal vein, there's another capillary. And this is meant to represent a capillary, a lymphatic capillary. And the lymphatic capillary is known as a lacteal. So that's the overall organization of this villus. The thickness of this villus is only a few micrometers. And then over the top of this entire structure, I'll show this in orange, is the so-called unstirred layer. Now, to most of us, this isn't intuitively logical. The idea that you have this washing machine out there sloshing fluid back and forth and back and forth, but yet once we get down to 30 or so micrometers above the apical surface of the epithelial cells, we now have an unstirred layer where the fluid is really quiet. Well, but to those people who have had some background in fluid dynamics, they'll recognize this immediately, that anytime you put fluid in a tube, the layer of fluid, the infinitely small layer of fluid lying along the wall of the tube, doesn't move. And the closer you get to the wall, the less and less movement you have. So for our purposes here, once we get down to 30 or 40 micrometers, there's almost no movement in this. And so what we find is that movement of small molecules, in the unstirred layer is by diffusion. Simply the idea of diffusion we've talked about over and over again in here, the idea of the thermal activity driving these molecules moving randomly in that unstirred layer. So what I want to do here, though, is I want to take a small subset of these enterocytes, these epithelial cells, and lay them out side by side and try to organize them with respect to the underlying capillary of the hepatic portal system. So what I'm going to draw here is a series of boxes, and each one of these boxes, of course, is going to represent an enterocyte. So each of these is an enterocyte. And the enterocytes, like all epithelial cells, sit under basal lamina. So they now have a basilar surface. But then, as with all epithelia, they also have an apical surface. But we find in these enterocytes that the apical surface has been thrown into extensive folds. Each one of these enterocytes have their apical plasma membranes thrown into these folds. These folds have a name. They're called microvilli. And the microvilli are just extensions of the enterocyte plasma membrane. And then immediately above these microvilli is this structure in orange, which I'm going to label here as the, let me extend it out a little more. This is going to be the 30 to 40 micrometer thick layer known as the unstirred layer. Well, one of the things we're going to find is that we're up here in the bulk phase where we have converted starch into maltose, that this maltose will eventually find its way down here into the unstirred layer. Maltose cannot be taken up into these epithelial cells. Maltose is out in the unstirred layer, so we have to do something with it to bring it into the epithelial cells. Well, there's an enzyme out here called a brush border. The brush border enzymes are transmembrane. Oh, I wish I hadn't done that. The brush border enzymes are transmembrane proteins. So one of the major players out here, and I'll put this in black, it's going to be a transmembrane protein here called maltase. Now remember from our early discussions that maltose is a disaccharide. So maltase is a disaccharidase. And what this does is in the unstirred layer, it finds a molecule of maltose and converts the molecule of maltose into 2 molecules of glucose. Now then, across this membrane, I'll put it in brown, across this apical membrane now is a molecule called a sodium-driven co-transporter. The sodium-driven co-transporter has the capacity of, on the apical side, binding an ion of sodium and a molecule of glucose. And when this transmembrane protein binds an ion of sodium along with glucose, it effectively moves the sodium into the cell. And it moves the glucose into the cell. Where have we seen this before? Proximal convoluted tubule. It's in fact the same set of genes. Identical genes here working in the epithelium of the small intestines we saw in the proximal convoluted tubule. Similarly, once the glucose gets into the cell, there's an avenue out here by GLUT2. And you remember GLUT2 is a facilitated transporter which simply allows glucose to diffuse down its concentration gradient. And from the image here, recognize that there will be the capillary. The capillary of the hepatic portal vein. So that once the glucose goes across the basolateral membrane, it now can diffuse into the blood vascular compartment, and that will be swept off to the liver. But again, this is the same thing as we saw in the peritubular capillary and the proximal convoluted tubule. Same story. Similarly, remember in the bulk phase up here, one of the other major players is trypsin and chymotrypsin, converting proteins, these great big proteins, into polypeptides, into peptides. So what will happen then is the peptides, I'll put them over here, which may be 20 or 15 or 30 amino acids long, where this is a peptide, on the brush border of these enterocytes, on their plasma membranes, will be a transmembrane protein which is known as an amino or carboxy peptidase. Well, this should remind you of the fundamental structure of all proteins and peptides. Remember that they'll have a carboxy end and an amino end. And what this enzyme will do is it will recognize either the carboxy or amino end of the molecule and clip off an individual amino acid. So where the bulk phase has generated proteins into peptides, the peptides find their way into here, and now the final phase is to cut these guys into individual amino acids by nipping one amino acid at a time off of their ends. Then, as we saw before in the proximal convoluted tubule, there's also a sodium-driven amino acid co-transporter. So that again, when we bind an atom of sodium out here, an ion of sodium, along with an amino acid, then the amino acid and the sodium are transported into the cell, and then across the basolateral surface, there is an amino acid facilitated transporter, which now allows the amino acids to leave the cytoplasm, the intracyte, go out into the extracellular space and enter the capillary of the hepatic portal vein. So we're going to leave for the time being the lipids in the background. And we're going to see that we're going to use lipase to do something a little different a little later. But we have to bust up those big globs of fat using bile first. Yes?
What was the purpose of the brush border enzymes again? What was their function?
So the question is, what is the function of the brush border enzymes? The brush border enzymes are a whole cohort of transmembrane proteins that do the final cleaving of molecules into molecules that can be transported across the membrane and into the intracyte.
The enzymes we're learning about are all they're individual classifications of brush border enzymes?
So they're bulk phase enzymes that work in the lumen of the tube, and those are released by the exocrine pancreas. Okay, with that. Now we're going to have another subset of enzymes that are called brush border enzymes, and they're transmembrane proteins found in the microvilli. Now I should point out here, and this is a question that I think brings this up, is that each of these cells is tied to its neighbor by tight junctions. And that when we talk about the microvilli, we're effectively talking about the extensions of the plasma membrane of individual enterocytes. But when we look at the enterocytes taking an aggregate, we talk about the combination of all their microvilli as being the brush border. Because to the early microscopists, these were all done by anatomy, and to the early microscopists, this looked like a brush with all these little fingers, so they called it the brush border. And these enzymes are so-called brush border enzymes, but when you hear someone say it's a brush border enzyme, it's just a transmembrane protein. Yes?
What happens to the sodium after it's taken into the cell with glucose or amino acids?
So what happens to the sodium? Well, you can recognize they're going to be filling up these cells with sodium. Down here, there's going to be a sodium-potassium ATPase. So as we fill it up, we pump it out. Some cases, they're probably pumped out back into the outside too, because you need to keep a fairly high concentration of sodium in the extracellular space, or in the lumen of the tube, I mean. Anything else? Now you'll notice that I've left a couple of things out here, and you may recall, let me put them up here, that in addition to those bulk phase enzymes, there are brush border disaccharidases. These disaccharidases convert disaccharides into two hexosugars. Well, we've shown one here. This maltase is a classic disaccharide. It is a transmembrane protein that converts maltose into a molecule of glucose plus a molecule of glucose. But there are two other of these disaccharidases I want to tell you about, such that, let me go over to here, this one. And sucrose will come into the unstirred layer. And to remember, sucrose is made of a molecule of glucose and a molecule of fructose. So now, here, on the brush border, there's this transmembrane protein. This is called sucrase. And sucrase now converts sucrose into a molecule of glucose and a molecule of fructose. So now out here in the unstirred layer, right beside the enzyme, we have a molecule of glucose and a molecule of fructose. Again, as a transmembrane protein here, and I'll put it in red, this is going to be a sodium-driven co-transporter. So we'll bind, just as we did here, with the products of maltose. We'll similarly use this transporter to snag a molecule of glucose, an ion of sodium, and bring the glucose and sodium into the cell. Same transporter. Now, truth of the matter is, fructose is transported by a different transporter. It's called GLUT5. It's a special protein. It's different. That's all you need to know. Let it go at that. And take a biochemistry course and learn more about it. Then here on the cytoplasmic side, there is now GLUT2. And there's also going to be GLUT5, which is the fructose transporter. But in the end, we're now moving these hexosugars across the basolateral membrane and into the capillary of the hepatic portal system. So one more character I want to tell you about, which I find the most interesting. And out here, I better not do it. I'll put it in green. This is a brush border enzyme, and this enzyme is lactase. Now, lactase, of course, is unique to mammals. After all, mammals are the only vertebrates that generate milk. We name the class Mammalia after mammary glands because they make milk. So all mammals, all mammals before they're weaned will have the capacity to digest lactose. So lactase converts lactose into a molecule of glucose plus a molecule of galactose. Now, if you look at the structure of glucose and galactose, they're remarkably similar. The only difference is that the number 4 carbon is the position of the hydrogen and hydroxyl with respect to one another. So they're really similar molecules. So what lactase does out here in the unstirred layer is it generates a molecule of glucose and a molecule of galactose. And both glucose and galactose can now be transported across the brush border membrane, across the apical membrane by a sodium-driven cotransporter. So in each of these cases, we have a disaccharidase, which is a transmembrane protein in the microvilli that converts the disaccharide into a monosaccharide, the two molecules composing the disaccharide. And then we use sodium-driven cotransporters to bring those sugars into the cell and then again use a facilitated transporter to take it across the basolateral side and into the hepatic portal system. Now, the reason that this lactase rather, is my most interesting of the enzymes is because it has a really unique human story. So again, if you're a mammal, once you're weaned, you no longer need to digest lactose. You'll never see it the rest of your life. But what we find is that if we look at the genetics, that it appears that in Western Europe, probably right now, what's the area of what present-day Ukraine, out on the plains in that area, there were these gigantic cow-like creatures called aurochs. And it looks like about 7,000 years ago, maybe 8,000 years ago, a group of people who lived in that area domesticated the aurochs and brought them basically into the backyard. And they, if you put your phone away, I'd appreciate it. And then in the backyard, they could go out now and again and basically slaughter one of these animals because it was a hell of a lot easier to do that than to go out and hunt them. So for at least 1,000 years, maybe a little more, they simply had these animals as meat animals. And then somebody had the bright idea, well, if a cow feeds its calf milk, maybe we can use it too. And so they started drinking milk. But for the most part, it didn't do them much good because they were normal mammals. And they had inactivated the lactase gene when they were about 3 or 4 years old, when they were weaned. And so they no longer contained the lactase enzyme in their brush borders. So basically, the lactose passed through the small intestine, went to the large intestine, and made terrible amounts of gas. But if you now look at the population of Northern Europe, those areas in Sweden, Finland, Northern Germany, England, 90 to 95% of the population retains the lactase gene. And if you look to see what happened, you find that there appears to be a single mutation some 6,000 to 7,000 years ago. One individual had a change in one base in the upstream regulator. When there was one base change in the upstream regulator, it no longer inactivated the generation of the lactase gene. It no longer downregulated the lactase gene, and it remained on. These individuals then were able to drink raw milk. They could then take in raw milk, get the protein out of the raw milk, and also get the calories out of the lactose. Apparently, it's provided a tremendous selective advantage. It looks like that in a matter of 1,000 years or so, within 10 or maybe 15 generations, there was a sweep of this gene through that population, such that almost all of those individuals could digest lactose. Now, interestingly enough, if you look in Europe and you look at sort of dairy culture in Europe, the northern portions of Europe will commonly use raw milk. But as you get farther and farther away from Northern Europe, you find less prevalence of the lactase gene in the population. And then what you find there is more and more fermented milk products. So now you're starting to make yogurts and sour creams and cheeses. So the fermentation converts the lactose into glucose and galactose. And then you can handle it. So this would still be just sort of some odd sort of case of human history. But wherever dairy has come up, whether it's in sheep in areas around Pakistan, whether it's in camels in what's today's Saudi Arabia, or if it's in East Africa with zebu cattle. In those cases, as recently 3,000 to 4,000 years ago, wherever the dairy culture has come up, there's been a mutation in the upstream regulator that turns off the lactase gene, so that lactase remains in the population. And what you find is it's different sites in the regulator gene, that it's not always the same base change. There are a number of different bases you can change in that upstream regulator, and it'll turn it off, so it doesn't function properly to turn off the lactase gene. So that appears that it's arisen several times in human history, and in each case, within a matter of maybe as few as 100 generations, we see that gene sweep through the population because of the tremendous selective advantage of being able to digest milk. I suspect when we start getting into the genetics of this, we're going to find that with the rise of agriculture, there are whole new sets of genes that come on that allow people to live in an agricultural society. In fact, you see some of that with the pancreatic amylase. You see when we start eating lots of grains that you get a proliferation of the amylase genes. But I find this sort of interesting because when I was in college, we were told that humans didn't really evolve after the last 60 or 70,000 years or so. That once people started walking out of Africa, there was no more human evolution. But clearly that's not true. We have a number of these examples where there are selective pressures placed on human populations, and those selective pressures will act just as they do throughout evolution and select certain individuals for survival. But with that, let's then think a little bit about the fate of the blood that's in that hepatic portal system. So let's take this panel and talk about what's going to happen to the blood that is flowing through the hepatic portal vein. So if you look at that upper panel, you'll see that the branch of the hepatic portal vein that lies immediately beneath the enterocytes is strategically located to take up, simply by diffusion, the molecules of glucose and the molecules of amino acids that have entered the extracellular space and then subsequently diffused into the hepatic portal vein. Now what we want to do on this panel is talk about the fate of the hepatic portal blood. And so I'm going to label this as the liver lobule and the fate of hepatic portal blood. Well, the first structure I have to tell you about is something called a liver lobule. A liver lobule is in the neighborhood of about 2 to 3 millimeters in diameter. So it's a multicellular structure. It'll be composed of thousands and thousands of these of individual hepatocytes. But if you look at a liver lobule, one of the striking characteristics of it is that it is a hexagon. And at each of these vertices, we see there is the confluence of 2 blood vessels. One of the blood vessels, I'll put it in blue, is a branch of the hepatic portal vein. But running right alongside it is a branch, I'm going to show this in red, is a branch of the hepatic artery. This is a branch, this is an arteriole, if you will, of the hepatic artery. This is arterial blood. This is oxygen-rich blood. This is the blood that's providing the oxygen to the cells of the liver. The hepatic portal vein is carrying poor but nutrient-rich blood. The blood to the liver is in the neighborhood of about 30 to 50, probably less than 50% hepatic, 50% arterial blood from the hepatic artery. This is a branch that comes right off the descending aorta. It's highly enriched for blood, so you have fully saturated hemoglobin here, and that's providing oxygen-rich blood. But the other, 70 to 50% is coming from the hepatic portal system. And it's emptying into this structure. Now, if we look at this thing, each of these tubes empties into a structure known as a sinusoid. Such that this region in here is said to be the central vein. But each of these green structures I've drawn here that's carrying mixed oxygenated and deoxygenated blood has a name called a sinusoid. This is just a fancy name for a liver capillary. And like all capillaries, this capillary is going to be lined with an endothelium. So we have this continuous layer of endothelial cells. So the liver capillary is lined, I actually should say formed, by endothelial cells. As always, these endothelial cells have an orientation. There's an apical surface facing the lumen of the tube, the lumen of the sinusoid, and a basilar surface that's facing the underlying tissue. And what I want to draw in here is that right on the other side of those endothelial cells are these boxes. I'm putting it in brown. And these boxes are hepatocytes. The hepatocytes are effectively liver cells. Now remember what's going on here. Really? Okay. Remember what's going on here. There's blood flowing by these cells, and what these cells are going to do is they're going to take up. They're going to take up amino acids and glucose. And they're going to convert the amino acids back to protein, mostly albumin. And they're going to convert the glucose into glycogen, which is basically animal starch. It's a branch structure of glucose. But there's another thing these cells do, these hepatocytes. These hepatocytes synthesize and secrete a detergent called bile. Now, I will never hold you to the structure, but let me remind you of the structure of glycerol, the structure of cholesterol. This is cholesterol. It's hydrophobic. And out here in the number, under this D ring, there will be a taurine or glycine. These are charged. So now you generate an amphipathic molecule. This is a detergent. And its function is to emulsify fat. Give me a couple minutes here. So here in the backboard, we have this fat globule. Bile breaks this beach ball-sized structure up into a set of BBs. And these are fat droplets. And these fat droplets now, because what we've done is we've increased the surface area to volume ratio, these now can be attacked by lipase in the bulk phase. And the lipase converts triglyceride into a molecule of monoglyceride and 2 molecules of fatty acid. These now are in structures known as micelles. So we have these micelles that contain the monoglyceride and fatty acids interspersed with bile. These micelles now can approach the brush border in the unstirred layer. The micelles now can have the monoglycerides and the fatty acids cross right into the cell. And then in the ER, in the smooth ER, the monoglycerides and fatty acids are reconstructed to triglycerides. And then those triglycerides in the Golgi apparatus are complexed with protein to make something known as a chylomicron. Those chylomicrons are released from the basolateral surface. They then get into the lacteal and go out in the lymph. And with that, I'll let you go.